Hello, everybody, and welcome to today's webinar. My name is Burke. I'm a Scientific Training Officer here in the training team at the EBI, and I will be your host for today. So I'm going to hand over to Elash, who's sitting just beside me at the moment, and we'll get started with today's webinar.
Okay. Hello, everyone. I'm a first-year PhD student here at the EMBL-EBI in Alex Bateman group. And for the first part of my PhD, I've been looking at detection and the study of evolution in repetitive proteins. And I'm going to talk a bit about it today. So the outline of this webinar would be, at the beginning, I will explain a bit. I will try to convince you why protein repeats are important. Although I think if you turned up for this seminar, you are already half convinced, so it should be easy. Then I will explain what are the tools and methods we can use to detect protein repeats. And the third part would be to analyze duplication patterns from these repeats in order to understand what were the evolutionary mechanisms that led to these proteins. And throughout the webinar, I will be showing an example protein in order to demonstrate what I mean with the concepts and tools. So first of all, a little bit of a small task for you. So what can you say about this protein sequence? I'm representing it at the top as a black line, the extent of the protein sequence, and at the bottom you can see the actual sequence. Probably by the title of the talk, you already know that there will be some repeats in here, and I will try to help you and tell you that this is one domain, and maybe you spotted that there's even another domain which is of the identical sequence than the first one. And there are even further two more domains which have 100% identical. So this schematic I will be showing throughout the webinar, which will be the protein sequence in the background as a dark line. And then on top, I will show these colored boxes for each of the domains or repeating units that can be identified. So this is just a made-up example, but actually, tandem repeats are quite frequent in proteins and also throughout all kingdoms of lives. So for example, we can see that in archaea and bacteria, there's about 6% of all proteins that contain repeats, although from these repeats, they are rather low copy number. But when we go to eukaryota, and especially mammals and higher eukaryota, we can see that the number of repeats in tandem increases. And this has been associated in many studies that the complexity of the organism can be also correlated with the number of tandem repeats that the proteome of the organism contains. And this can be explained by different aspects, but one of them is the functional relevance that these protein tandem repeats have. And one of the most important functions they have is to be involved in transcription factors and protein-protein interactions, and also to function as adhesions. So they are really important in signaling and other complex cellular functions. And so just to give you an example, this is the epithelial growth factor transcription factor, and then we can see that the DNA binding region is formed by 3 tandem zinc finger domains, and each of the zinc finger domains, which are here represented as these 3 different colored boxes, has a different specificity for a DNA sequence. So together, when you combine these 3 different repeats, you get a long DNA, very specific sequence that the protein is recognizing. In addition, a bit more relevant in bacteria, these tandem repeat proteins provide an easy mechanism for the organism to adapt. So, for example, in this Staphylococcus aureus, the cell surface protein RIP contains up to 10 or 11 tandem RIP domains, and they are highly identical, so that the bacteria, through some DNA recombination and other molecular mechanisms, can very easily change the length of the protein and effectively the number of repeats that these proteins expressed at the surface contain, so that it can evade easily the immune system by changing the aspect. So this was seen in one recent study, that compared GBS strains which were carriage or diseased, and what you can see in this box plot is that the diseased, the strains that caused disease had a lower number of tandem domain repeats than the strains that were non-pathogenic. So we can see another advantage of having proteins that contain repeats. So the example I'm going to be giving throughout the webinar would be a protein that contains a domain which is annotated as domain of unknown function, 1220, and this DAF1220 has been reported to have a striking human lineage specific increase in copy number. So what this means is that if we look at the number of domains that throughout the evolutionary tree, we can see that species which are closer to human have a higher copy number than species which are farther away. And we can see that chimpanzee has less than half the copy number of humans, and orangutan has even less. And mammals some other mammals still have a few of these domains, but none is found outside of the mammalians. And furthermore, this domain has also been associated with brain, so including brain and expression in neurons, which could be an interesting target in order to do a study of tandem repeats in proteins. So if we take a look at the PFAM entry for these domains, we can see, first of all, in the species tab, we can see that this domain is only found in Eukaryota, 100%, and then the species that has the most domains is Homo sapiens, with 73 sequences. But we can see that other primates also have proteins with a domain. And then we go back to mouse and other species down here. And then another thing we can look from the PFAM information is the domain organization of these proteins that contain the domain. And what we will see is that although a few sequences only contain one domain, there are many sequences that contain repeats, tandem repeats of this domain. And for example, in order to demonstrate the concepts in this webinar, I selected this protein, which is the neuroblastoma breakpoint family member 14, which has about 30 tandemly repeated DAF1220 domains. And if we click at this protein, and then we even further go to the UniProt entry, we can download the sequence as a FASTA file. I will repeats. And, so I will download it and store it here just to use it later. So I already have it, so we won't download it. Okay, so we've identified one protein that contains tandem domain repeats, but then we want to study it further. So this is the protein we've just seen. I will skip through. So once we have this protein sequence, how can we identify the repeats automatically? Because you have seen that identifying them by eye was a hard task, and we don't want to do that for millions of proteins. So the easiest thing we can do is just compare one protein against itself. And then what we should see in this comparison, we will see a pixel of higher intensity when two amino acids are identical. And what we can see is trivial here, which is the first domain corresponds exactly in the first diagonal. But then when we will see when we extend this analysis further, would be that off main diagonal lines will appear, and those correspond to those will correspond to the first amino acid in here being identical to the first amino acid in here. So they will identify regions of the sequence which are far apart, but they are identical to each other. So the longer these lines are, the longer the repeats, and the more lines we see, the more repeats we see in tandem. So there are a few resources to visualize and calculate these dot plots. So there's one which is available from the Sanger website called Dotter, and there's a JavaScript one which can be used online. But I will take a look. So I will use the desktop one, which has to be installed, and we just have to pass the same sequence twice in order to obtain the dot plot we are interested in. So this is the neuroblastoma protein family 14, and in the dot plot we can see that the first part of the sequence contains a duplication here, and after that we can see that there are many diagonal lines that correspond to repeats, to tandem repeats between, and they go all along until the end of the sequence. So if we close that, so we have identified these repeats from the sequence, but what we want to further analyze what's the similarity between them. And so we've done this part. So that there are a few methods that from these dot plots, self-comparison dot plots, they can extract the repeats, and because they will just try to reconstruct off diagonal lines and build consensus sequences out of this. So a few of them are radar, Swelfe, and T-Rex. And all of this would work just fine, but for our example, we have additional information, which is that each repeat corresponds exactly to one domain, and this domain is already annotated in PFAM. So instead of doing de novo repeat detection, which would be the use case for those methods, what we can do is we can just extract from the PFAM alignments of all the domains, that 1220 domains. We can extract the alignments, and then we can just select those domains that appear in this protein in order to compare the similarity between these tandemly repeated domains. So I'm going to do that now, and so I will go back to the PFAM family .1220 website, and then I will go into the alignments tab, and in here we have one section which is format and alignment, and this will contain the alignment of all sequences identified, all domains identified as belonging to this family. So what we can do is we can take the full alignment and format it in Stockholm, and also sort it alphabetically, and we can just view the alignment first. And what this will generate is a rather big file with all the sequences and the regions of the domains from this family. And then down here we will have all the alignments, which is the interesting part we have. So if we go ahead and save, so that was repeats webinar data. So if we go ahead and save this alignment as protein family 06758, which is the PFAM code, as full, and then give it the STO extension, we will be able to open it later in a sequence alignment viewer. And so this is what I'm going to do now. So I'm going to take this file I downloaded and input the file into JALView, which is a sequence viewer and multiple sequence alignment viewer. So I'm going to select the Stockholm format. Oh, sorry, I forgot one thing before that. So we have, if we look at the alignment here, we have downloaded all the sequences all the proteins that belong to this, all the domains from all the sequences that belong to this domain family. But what we want is only the domains that appear in our sequence. So in order to do that, we can just use grep in order to extract the domains that belong to the sequence we're interested in, and then copy this result into a new file. So I will do that. So I will do grep and then the name of the sequence, and then we have this file we just downloaded, and I'm going to copy it into a new file which will contain only the domains from this sequence. And that's the file I'm going to open with JALView. So I'm going to select Stockholm format here, and I will open this file. Okay, so this file contains a lot of gaps, but it's because it contained we can just remove the empty columns that were left before. We'll have a much compact alignment, and then we can also color by cluster to have a nice overview of the similarity between all the repeats. And I'm going, because these were sorted alphabetically, I'm going to have to move these few sequences on top in order to have them sorted in the sequence. So now you can see that the indices are sorted sequentially by sequence. Okay, so we have our multiple sequence alignment loaded and visualized. So what else can we do now? So a very easy and useful analysis we can do is to analyze the similarity patterns that exist between these repeating units, or in our case domains, because we can infer some evolutionary events out of these patterns. So in this case, it's depicted here a protein with 5 domains, and at some point in evolution there was a tandem duplication where domains 3, 4, and 5 were duplicated. So these 3 domains were duplicated into 6, 7, and 8. And this triplet duplication can be still observed because domains 8 and 5 are similar to each other, 7 and 4 are similar to each other, and 6 and 3 are similar to each other. And this can be better seen with the the sequence similarity matrix, where we can identify as well, like in the dot plot, of diagonal self-diagonal diagonals that correlate domains identical to each other. And another plot which is useful is the autocorrelation vector, which simply calculates the average sequence similarity of domains at a given distance. So in this case, domains that are three domains distance apart have the highest sequence similarity when compared to the other ones. So for example, 3 and 6 will have the maximum highest similarity, 4 and 7 will have the highest, and 5 and 8 as well. So we can do this. So this is just another way to see how the colors in this sequence similarity matrix represent the similarities between the domain repeats. And if we apply this analysis to our protein, a nice pattern is revealed, which is the same as the one in the other protein, which is that every 3 domains, so the duplication events were coming from 3 domain units, and these 3 domain units can be seen by the maximums in this autocorrelation vector plot, and they can also be seen by the diagonal matrices in the matrix. So this was our alignment, and that we opened in JALView. And what we can do in order to further see the duplication patterns would be to calculate a tree out of the, so we have to select all the sequences. We have to select all the sequences, and we can calculate a tree out of this. And then the tree will just represent the different similarities between the domains. So we can trim the tree at different levels. So what we can see here is that there are 3 very marked blocks, and if we go a bit further, so if we go here, we can see that the first, maybe it's not that clear now. So the first 3 domains are rather different to each other, but after that we can see that every 3 domains the color patterns are maintained, and this clearly means that domain starting at position 600 corresponded to domain starting at position 800, and so forth, and there were at least 1, 2, 3, 4, 5, 6, 7, 8, and possibly 9 duplications in this protein in order to achieve the complete one. So if we close this one, go back to the presentation. So what we've seen is that this protein, the repeats in this protein were formed by a 3 domain unit repeat, and we can take these 3 domains that were repeated and construct our repeating unit, which will consist of the 3 domains. We can go ahead and look for all other proteins that contain this domain of unknown function, whether the repeat unit of 3 domains was conserved or not. So what I will do is I will open a hammer job, and then I will open the, so I have extracted this 3 domain repeat. I extract the 3 domain repeat and called it Yeah, called it like this. So I will just try to search for this sequence, and then I will select all UniProt and then submit. Should be relatively fast. So we have our results. So this 3-domain repeating unit was mainly found in Homo sapiens, which we expected, but we can look at the taxonomy in order to get a better understanding, and we can see that mostly, so there are about 200 primates that contain this one, and it's mainly mammalia, as we expect, and we even have the count of the count of proteins for each species, and of course it's higher in human because we knew that when we started with. But the interesting part would be that if we look at the domain organization of the protein hits we got, what we will see is that the proteins we obtain are all, so can all be explained by this 3-domain repeat unit. So as you can see, there's the proteins are have increasingly more repeats, and all these repeats we see are coming from this 3-domain repeat unit, which is a strong suggestion that the evolutionary mechanism behind the generation of all of these longer proteins is the same one as the one we saw in our study protein. So we've come to the end, and as just a quick summary, I've tried to convince you that tandem repeats are important for protein function and evolution, and that there are also a few tools to detect and visualize tandem repeats, and that these evolutionary events that caused the tandem repeats in proteins can be inferred from their sequence similarity patterns. And identifying these recurring protein repeat units across many different proteins can be is an important source of information or evidence in order to go ahead and study what are the molecular mechanisms or the functions that these units represent or carry out. So this was the end of the talk. I just give a few, if you want to contact me in order to discuss any of the topics I've covered, here is my email, website, and Twitter handle, and I'd be happy to take questions.
So if anybody has any questions for Alice, you can write them into the chat box, and we'll just give you a minute or two to do that. While you do so, just a reminder that we are running a series of webinars, and there are lots more coming up before we break for summer in August. The next one is on the protein database in Europe, and they've had lots of updates recently, so that would be a great one to catch if you are working on proteins. So I can see that there is a couple of questions coming through now. Perhaps we can start with the one about, what about mutation rate of tandem repeats? How can you go about measuring that?
Yeah, so one thing you can do is from the alignment we've seen. So from the alignment of all repeat units. So in our case, the three domain repeats were quite conserved throughout evolution, but in the case that, so it's very hard to distinguish whether the domains in here came from a duplication. So we will assume that it was a duplication of 3 domains because this is what the whole family seems to indicate, but they could also have come from a duplication of 6 domains or 9 domains, right? Any multiple of 3, because from the duplication patterns we couldn't distinguish it. And in case there were some different mutations, what you could infer from the alignments would be that the mutations would cause some functional implications, and then those would be the so you would have an interplay between the neutral mutations that would happen throughout the evolution of this protein after the duplication events happen, and those mutations that really were crucial to distinguish the function between the different repeat units and therefore affect the whole function of the protein. Not sure if that answered your question. So that was the first one.
So the next question is about, it says, to show that the 3 repeat unit is fundamental, wouldn't you have to look for hits that don't come up in the 3 unit hammer search?
Yeah, that could also be an option. But what I'm trying to demonstrate with the 3 unit repeat is that there is some underlying molecular mechanism. It's either an exon organization, or maybe it is some sequence that causes DNA slippage, some kind of molecular mechanism that makes this 3-domain repeat the one we see most frequently. But it's true that we can assume that this domain appeared maybe in isolation first, and there was a first duplication event, which I haven't looked throughout thorough enough to see whether there's enough sufficient sequences in order to see what, because they might have diverged so much that there is no signal, no signal so far. Because, for example, one thing you can see is from this unit of 3 different repeats. So, for example, if we try to align the first one and the second one, we can just do a pairwise alignment, and they will be 71% identical. And then so yeah, we can do the second and the third one, and they will also be 71% identical. But we cannot relate which one was the original, and because we only have three domains.
Okay, so there's also a question from Diego. How about duplication and deletion rate versus point mutation rates? Is there a way that you can look at that?
Okay, yeah, so I see what you mean. So I assume you mean by duplication or deletion of whole domain units. So yeah, from this is rather a simplistic view, the approach I've taken, because I have assumed that from my duplication matrix, from my sequence similarity matrix in here, I have assumed that if there was a duplication, all the domains still were maintained in this protein afterwards. But it's true that there can be domain losses, and then those domain losses will probably be misinterpreted in this matrix. Whether you can, the thing is, so both things are quite interdependent in this analysis, because if the mutation rates are higher and the distance between the domains is also very high, very large, then the signal bit of the evolutionary relations is lost, and then the deletions of domains might not be assigned correctly. But I guess one could use this approach in order to see for one family in order to see for one family how diverged are the different duplications compared to the deletion, and then calculate the rates. But I haven't done that so far.
So are the sizes of duplication units conserved in different families?
No, they are not conserved. So some families have a very clear pattern, like for example, if you look, so here I had some extra slides. So in this family, if you look at the duplication patterns of many different proteins, you can see that they are almost all of them 3-domain repeats. But this is also maybe this family, because it's only found in a very small subset of all the organisms. It's also true, but it doesn't mean that these domains can be repeated in doublets or single domains. So, for example, if you look at the other families like fibronectin, or you look at nebulin, I think nebulins are duplicated every seven domains, and then fibronectins have a mix, but some proteins have a very strong signal for 2-domain repeats. I think it depends, and it's not conserved. I wouldn't say that.
Okay, thanks, Alish. And one last question: is there an objective way to define the phase of a fundamental repeat in multiple sequences?
I'm not sure what you mean about phase, but maybe you mean here the domain boundaries or the repeat boundaries. But this is clearly an issue when looking at protein repeats, because most programs that identify repeats de novo, because usually the termini of these domains are more divergent than the center of the domains. What ends up happening is that the repeats that are identified start at the medium, so at the middle of one of the domains, and then the whole units in the alignment you get at the output. So the alignment, yeah, it's maybe too far back, but the alignment you get at the output starts at the middle of one domain, contains the linker between the two domains, and then continues up to the middle. This is why using databases of known domains is a very useful thing. So if the repeat you are trying to analyze is annotated in a database of domains, there is a high chance that the boundaries are correct, so that the repeats you are analyzing actually correspond to functional and structural units. The protein interior or not. So actually this domain of unknown function I was looking at does not have a known structure, which makes it even more interesting to study. But it depends very much. But I think, for example, in the cases I've described, because the repeats correspond to full domains, they are usually independently folding and independently functioning in theory. So this would mean that so they in any case they just interact with other proteins, but they form on their own a globular structure. So the repeats contain the whole thing, though they are not in the inside or outside.
Okay, so we're going to have to leave it there for today. Thank you very much, Alish. That was a really good webinar. I'm not a protein biologist, and I definitely learned something from that. Otherwise, we've got some webinars coming up again soon, and you can see the full listing on our website. Thanks very much for joining us today, and enjoy the rest of your day. Bye.
Thank you. Goodbye.